not actually what Tasmanian devils look like. This is what they look like.
Oh.
Tasmania, which is down here off the southern end of Australia. And just some background on this species. So first of all, Tasmanian devils, because of their demographic history, have generally low genetic diversity. So for instance, here's a paper that came out a few years ago based on whole genome sequence data for two individuals named Cedric and Spirit, from right there and right there. Quantifying genetic diversity overall, you can see where this is where Tasmanian devils are. This is European humans, African humans, pandas, American bison. So you can see in the grand scheme of things, they have low overall genetic diversity, but not zero genetic diversity. So there is some population structure. This is mitochondrial haplotypes. There's kind of an east-west split, more or less. But in 1996, there appeared this strange disease that first appeared in this Mount William population up here in the northeast corner. This disease called Tasmanian devil facial tumor disease, or DFTD. So it first appeared in 1996 and started spreading rapidly. And you can see the advancing wavefront here. So in 2000, it covered this area, 2005, this, 2010, 2015. Within the next couple of years, almost certainly it will have covered the whole range of the species. So as the name suggests, this devil facial tumor disease is a set of tumors that grow mostly on the faces of these animals. It's a really nasty thing. And for the most part, animals who get this disease, who are infected with this disease, die within about 6 months. So that's been the general pattern over the past 20 years. Here's a paper that figured out that this cancer is actually a transmissible cancer. So that means it's the actual tumor cells that are transferred from one individual to another. And they figured this out because if you take samples of tumors from multiple different devils, they had very similar karyotypes, and karyotypes that were entirely different from their hosts. And if you look closer, if you pick up a devil who's infected with the disease, whether it's a male or female devil, if you sample the tumor will have 2 X chromosomes. In other words, the first individual that developed this cancer was a female, and then that cancer spread from that one individual to other individuals. So this is one of only basically 4 known transmissible cancers in the animal kingdom. One is in dogs, which has been around for thousands of years. It's sexually transmitted. One is in clams, which was just discovered the year before last, I think. Clams in the Atlantic Ocean. And the 4th one I'll tell you about right at the end. Okay, so this disease is a really nasty disease. It's spread throughout the population, so this is incidence of the disease in these different populations. The arrow shows, so each column is a population. The arrow shows when the disease first appeared. One thing that's interesting is that they transmit it by biting each other, and they basically only bite each other after they become sexually mature. So they become adults, and then they start biting each other during mating behavior and during feeding. They're scavengers, so they come together on dead wallaby carcasses and stuff like that. But they only bite each other as adults, and the disease is not transmitted from mother to offspring. And so you can see here, these are disease incidents in 1 to 2-year-olds, 2 to 3-year-olds, and 3+-year-olds. And you can see it's basically the individuals that are 2 or 3 or older that get the disease. And the young ones basically don't get it. A few in that population, but for the most part they don't. It's caused rapid declines in populations. So again, here the arrow is when the disease first appeared in these different populations, and you can see the numbers drop off. This one, if you notice, this population didn't really drop off. The interesting thing about this one is that this tumor cell population, this transmissible cancer, is evolving and changing also, and changing into multiple different strains. So this was actually a tetraploid strain of the cancer that hit this devil population first, and for whatever reason it didn't have nearly as big an effect on this population. These populations were hit by a diploid strain, and they had this rapid population decline. So here's another interesting thing. Since it's only transmitted among adults, juveniles don't really get it. And in general, if the population is really diseased, individuals will reach maturity, maybe have one shot at reproducing, and then get the disease and die. Whereas in the past, they would reach maturity and have multiple reproductive years. So what it's done is impose life history selection on these populations. So here's the number of 3+-year-old individuals in the population. Black is before the disease and white is after the disease. And you can see after the disease, most individuals are younger than 3. So all the 3+-year-old adults have disappeared from these populations. And here's the percentage of 1-year-old females that are observed breeding. Before the disease, basically no individuals bred before age 2. But now there are a bunch of 1-year-old females that are breeding. So it's shifted life history in these populations. So for some reason, the immune systems of these devils are not able to deal with this cancer and not able to recognize it as non-self. So you might suspect that's due to the low genetic diversity that I mentioned. It turns out it's not, actually. So this is MHC diversity, and there is quite a bit of MHC diversity, suggesting that the immune system should be able to handle this kind of thing. It should be able to recognize it as non-self tissue. But it turns out that the tumors actually are able to downregulate their own MHC expression and also suppress the host immune response. So these tumor cells have evolved these mechanisms to be able to transmit from one individual to another. Okay, so the disease first appeared 20 years ago. The first models, as it was starting to spread, the first models predicted complete extinction of the species as a result. But it turns out that hasn't actually happened. So what we did, one thing we did, is to set out and ask whether there's any genetic variation for resistance to the disease in these populations. So here's this paper that just came out. We have 3 populations here, here, and here. You can see the dates when the disease first appeared. We have samples, because this is it was recognized as such a threat to the persistence of the species, got a lot of attention, and people started going out and sampling these populations. And so we have samples, tissue samples from these populations from before the disease appeared and after the disease appeared. So here are our sample sizes. Black is before the disease and red is after the disease in these 3 populations. So we're able to look over time at the genetic makeup of this population. So we did red, 294 individuals total across these 3 populations. Because of the low diversity, like I was saying, we calibrated our number of loci to account for the low overall diversity. So we used, even though it's a big genome, it's over human-sized genome, we used a 6-cutter, PST1, that one right there, which gave us about 800,000 loci across the genome. So lots of loci. From that, we still got only 90,000 SNPs after all our filtering and stuff. But we have these multiple time points sampled. So the idea is that we're looking for signatures of selection based on genetic changes over time, from before to after the disease. So here's the workflow, just to give you an idea. For that data set. Let's see, we have DNA, rad preps, pretty standard. These were done at slightly different times, so we had some paired end data, some single end data. Paired end, we were able to remove PCR duplicates, not on the single end data, but we all fed it in here. Called SNPs using stacks, filtered by things like minor allele frequency and observed heterozygosity to get rid of paralogs. And then we examined each of these three populations as essentially three independent replicates. And we did a bunch of other filtering and analysis. And essentially, here's what we got. So, we're looking for a couple different signatures of selection here. The first is, this is just allele frequency change. So just straight change in allele frequency from before to after the disease moved in. Then here, this RSB statistic is a metric for change in linkage disequilibrium. So I mentioned if selection acts on a particular locus in the genome, often the best signature that it leaves is an increase in linkage disequilibrium around that selected locus. And so that's what this is trying to pick up. And then also based on the allele frequency change, we can get a direct estimate of the selection coefficient. So a direct estimate of the fitness difference between one allele and the other allele at a SNP location. So we have all these different metrics across these different populations. If you can see these three colors, each of these dots is a SNP location within one of the three populations. And the result is that we honed in on two genomic regions. So this is one of them. One last thing I'll just point out down here, each of these little graphs is changes in allele frequency at one particular SNP for the three populations. So the three colors are the three populations. And you can see that they're really parallel in a lot of cases. So these all go up, these all go down a little bit, these go down. So if you think of these populations as evolving more or less independently, at least in that 20-year time scale that we're talking about, the fact that they all have moved in the same direction at these locations is strong evidence that it's not just random genetic drift that's causing these little frequency changes.
Sorry, how do you know the allelic effects on fitness?
So that basically is calculated from the change in allele frequency over time and an estimate of effective population size, which we calculated using LDN. So it's basically a really simple population genetic model of selection coefficient S acting on one allele at a locus over X generations. So these are our two genomic regions where we have this sort of composite of signals. And in both cases, we're able to focus in on a couple candidate genes. So this region had just one gene called cereblon. This region had these 6 genes here. All of these genes have some link to cancer or immune function in humans. And so that's one reason why they're interesting candidates. I think I mentioned earlier that we have a reference genome for this species, but it's actually pretty poorly assembled, so it's in 30-some thousand scaffolds. But nonetheless, you know, this and this are a couple scaffolds that are relatively big. They're not anywhere near chromosome size, but they're still big enough for us to map a bunch of loci on there, identify a bunch of candidate genes in that region. They're big enough to identify the extent of linkage disequilibrium, that kind of thing. So it really shows that even a poorly assembled genome can give you lots and lots of information that can be really useful. I mentioned the extent of LD, so this is LD decay for these three different populations. Sorry about the axis labels on here, but this is 1 megabase, 1.5 megabases. This is the LD decay with confidence limits. So it extends out, 100 kb or more, certainly. Just like I showed before, this is just a correlation, right? So this is just linkage disequilibrium in terms of correlation. And so if you figure it extends out 100 kb, and our markers are spaced more densely than that, then we're likely to pick up the signal of anything that's being acted upon by selection. Because anything that's responding to selection is going to drag along some region of the genome in terms of allele frequency. So these are our candidate genes that I mentioned have cancer and immune function, look interesting for different reasons, all except for this one, uncharacterized protein. I don't know. But again, most of these were annotated in the reference genome based on automated gene annotation sorts of algorithms that are out there. And the only reason we know about their function, we don't know about their function in Tasmanian devils, we just know about their function for the most part in humans, maybe in mice. But there's enough conservation of gene function across mammals that we can make some inferences from that. Okay, so I'll just talk about a little bit of the other stuff that we're doing. So we have an expanded data set from 7 populations now. Here, there, and there. And again, we have time course sampling from before and after the diseases moved into all these populations. And here what we're doing is rapture that you heard about this morning. So again, the basic idea is that you do a rad library prep to subset down to this set of loci. But then we have a set of probes to subset even farther down to a targeted set of rad loci. So in this case, we designed an array of about 16,000 loci. So they're based on candidate selected loci that I showed you before in that first round of sequencing. They're based on genes of known function spread around the genome, and then a selection of loci just sort of scattered around the genome to give us that background.
Were all the 15,000 loci in or near selected regions, or just a proportion of them were sort of candidates for being under selection?
Just a portion. So these 3 groups are basically exclusive. So roughly 5,000 loci that were candidates, about 5,000 that had hadn't been previously identified, but had some gene function that looked interesting.
How many little SNPs or red tags did you have to choose from to narrow it down to these 16,000?
It was, so I said 90,000 SNPs before. It would have been a bigger list. So we would have used pretty strict filtering to get to that list of 90,000 that we were testing for selection. Here we were a little bit looser in the total set of SNPs we were considering. So these were filtering based on genes and gene function. These we filtered down based on minor allele frequency and low missing data in terms of genotyping and the individuals we had already. So these were basically just generally well-behaved SNPs.
What minor allele frequency colors did you use?
That's a big question. Something like 0.2, something reasonable.
Did you also use the criteria of sort sort of evenness of spread or distribution across the genome?
Yep, yep. Yeah, so all of these we tried to spread across the genome. But again, we have the genome's in a bunch of scaffolds. So I think we said, you know, only one locus per scaffold. But even then, there are plenty of scaffolds that are not represented on this.
This is a really beneficial aspect of capture combined with RAD. If you start with this in mind, you can tailor the markers you capture based on, you know, your question.
Right.
In addition to this, you could have markers, you know, diagnostic or at a high frequency in a certain population. If you want to do assignment tests, hybrid. If we have markers that are diagnostic of hybrids or some other species, you can throw those on your array. It's a really nice, flexible system to be able to add capture to your RAD.
Yeah.
So when you're selecting your immune cancer function loci that you want to target, do you have any problem having them close to RAD? You know, because they have to be associated with that site, right?
Right.
Because I was worried about that, and I want to do self-incompatibility loci, but you have to start with them being associated with that type. So how do you have any trouble with that, or how do you go about doing that?
Yeah, so that's a really good question. So we, all of these loci are RAD loci.
Right.
So they're not in these genes necessarily. Some of them are, but most of them are not. They're just close by. Basically what we did was identify a list of genes that looked interesting. And then one got rid of the nearest red locus to each one.
And then how near?
So I mentioned we kind of, from the LD data, we were kind of thinking of
It's only
the
like
extent of
100
LD being like 100 kb.
Yeah. Oh, 100 kb.
Yeah.
Oh.
So it's big.
Oh, okay. Okay, got it.
Yeah. So I can't remember specifically what cutoff we used here, but
And
most
so
of
you
them.
use your reference genome for that, and
Yeah. Yeah. So these are loci within some distance and on the same site.
right.
Um,
And you didn't have any trouble, they're high density enough RAD sites.
Yeah.
To get something.
right. So again, we used PST1, which is a 6-cutter, which is expected to cut every 4,000 cases on average. Yeah. So we had a pretty dense set of loci to choose from.
Just another comment while talking about the flexibility of the capture step. You can have different tubes with different baits and use or mix or use the different tubes. And one example is if you wanted a mitochondrial marker occasionally for certain questions, you could have a separate tube, and companies will provide those with a lot lower concentrations of that bait because mitochondrial DNA is so much more abundant, for example. So you could have a system of tubes for different questions, numbers of markers, locations of markers, that sort of thing.
Right.
So they're making those baits pretty reasonably priced. with
Yeah,
the
actually, so
baits.
this is a MyBaits kit. The company is called Microarray MyBaits, and it's like $2,600 for capture reactions with 20,000 loci. And I think for $2,600 you get maybe 12 capture reactions. But one thing like Mike was talking about, so we shelled out $2,600 for this many loci, 12 capture reactions. We did 3,500 individuals, but because we were able to multiplex so much, we did 8 plates of 96 individuals per capture reaction. In other words, 3,500 individuals only used 5 out of our 12 capture reactions.
And then do you tile at all, or do you just have one, you don't tile?
No, so there was no tiling here, and in fact, I think we just, our RAD loci, we had 150 base pair sequencing. I think for every locus, we just went position 20 to position 100
Okay.
for 80 base pair rows. So like I said, we had this many individuals. If we were to do normal RAD, especially using the enzyme that we did, we would have been pooling 24 individuals per lane, so was that 150 high-seek lanes if we wanted to do normal red. So that's why we did this. And then the other thing, because we have collaborators who've been collecting so much great data on these individuals, all of these individuals, or most of these individuals, we have phenotype data also. So case control, meaning presence or absence of disease, age at first detection of disease, if they're captured multiple times, length of survival with the disease, again, now if they're captured multiple times, and tumor growth rate. So we're doing a bunch of stuff with this data set. We're testing for selection just basically as we did before. Although we can't use the LD-based tests for selection because now our markers are too spread apart because we have a lot fewer of them. But we're mapping loci associated with disease, and we're also trying to develop basically a marker panel that would tell you an estimate of the susceptibility to disease in a particular individual based on their genotypes at some set of loci. So again, here's some details of the analysis. We use some bits of stacks, bowtie, SAM tools, angst, which we used in this case because coverage was still pretty low. It was something like 5x. So basically with this Rapture protocol, there's an extra PCR step, so we had more PCR duplicates than usual. We also had a lot of off-target loci that somehow snuck through the protocol. So we ended up with a little bit lower coverage than we were expecting, but by using this sort of Bayesian genotyping program, we're able to get good genotypes across these individuals. And multiple other sorts of programs, testing for selection with a couple different things, mapping phenotypes. This program, Gemma. I'll just show you some of these results here. This is testing for selection. DCMS stands for decorrelated composite of multiple signals. It's just a way of taking these multiple statistics from multiple populations and combining them all in some sort of statistical way. There's a bunch of what seem to be significant SNPs and potential candidate genes associated with those. Like I said, we're also able to do association mapping with these 4 phenotypes. And we're actually able to explain a relatively large proportion of variance, of phenotypic variance based on some of these genotypes. So some of these numbers like 77%, 64, 69% of variance in phenotype explained by genotype at a set of large effect SNPs. You can see the confidence limits here are pretty large, but a lot of them don't overlap 0. So we're picking up on some genetic signals, some genetic basis for these phenotypes. And then this, we're interested in being able to predict the susceptibility of individuals to disease. So on all of these plots, I'll just zoom in on one, which is the most significant one. So here we have, this is for the phenotype age at first infection. So the age at which an individual first is detected to have the disease. On the bottom is the true phenotype for these individuals in days. So, 1,000 days, these are roughly 3-year-olds, individuals who got the disease at age 3. And here is the breeding value, which is the predicted phenotype based on genetics. So the black dots are the model fit. The black dots are all the individuals that were used to fit the model relating genotype to phenotype based on some populations. And then the red dots are another population of devils that was not used to fit the model, but where we tested the prediction across the species range. So the idea of all of this is that, the disease just appeared 20 years ago. So if there's selection in response to the disease, if there are alleles that make individuals better off in the face of the disease, those are not new mutations in the last 20 years, in all likelihood. They're not new mutations. There's standing variation that's been hanging around in the devil population. Maybe neutral or maybe not, but it's standing variation that's responding to selection. And so this is telling us that likely some of that standing variation is spread pretty widely across the species range. It's not likely to be confined just to certain areas. Okay, some new things. So some populations now seem to be recovering, including this one. Basically, this one that was in the study seems to be rebounding in numbers. So we're looking into that. There are some individuals that are showing regressed tumors. So they get caught with the tumor on their face and they get caught 6 months later and the tumor has shrunk or even disappeared entirely. And that's just in the past couple of years that's been observed. And then, I told you I was going to tell you about the fourth transmissible cancer known in the animal kingdom. It was discovered in Tasmanian devils. So here we have, out of 4 transmissible cancers in the animal kingdom, 2 have appeared in the last 20 years in one species. So it suggests something makes these animals particularly prone to developing transmissible cancer. And the interesting thing, I said that the DFTD, if you take a tumor sample, it has 2 X chromosomes because it came from a female. One reason we know this is the second one is it has an X and a Y. So this was a male that got this one. Okay, so we're continuing to look at genomics of the recovering populations. We're doing a bunch of whole genome sequencing. We're testing this hypothesis that devils are especially prone to this kind of disease, and maybe it's happened multiple times in their evolutionary history and left a signature, a longer-term molecular evolution signature on their genomes. And then we're doing some targeted sequencing of some of the candidate genes. Okay.
So this is all really cool, and in many ways it's often what we do in conservation, which is sort of just documenting what's going on out there, right? So you're documenting the recovery. Is there any use of this to identify individuals that are resistant and doing any breeding or anything? I don't
Yeah,
want to
that's a
breed
great question.
Tasmanian devils, but
Right.
so to do some active conservation.
Yeah, that's a great question. So when the disease first appeared, they started setting up captive populations because the prediction was they would go extinct in the wild and then they would just repopulate. And so this, I don't know if you can read this, this is one of the captive populations sponsored by some store chain called Chicken Feed, called the Chicken Feed Tasmanian Devil Sanctuary. So there are multiple captive populations. Part of our idea is if we can identify some genetic basis to resistance, you might want to manage for that in these captive populations. They're also doing some reintroductions from these captive populations, which may be a really bad idea if the natural populations are evolving some degree of resistance, and the captive populations, which have been disease-free, have been free to not evolve any resistance and have it be dumped back into the natural populations. So part of the message of finding evidence of evolution in response to the disease, part of the message is maybe we should let the natural populations do that instead of supplementing them with susceptible individuals from the captive.
Well, when they were taken, that was before they knew about any susceptibility, like differences. So if you're developing the panel, because like you're saying, yours, if it's in the last 20 years and the standing variation was there, so could you, I mean, because I'm assuming they're recovering, but the numbers are lower, you could say, well, these ones are okay. to outbreed and these ones aren't? Is that?
Yeah, so part of the idea of a susceptibility panel would be to test captive ones, because they probably have some of that variation in them.
Yeah.
We don't know.
And presumably have been fed and are, energetically robust to
Yeah,
reproduce.
yeah. All right, so I'll just show you a video here. So this is a graduate student from the University of Tasmania. And this is in the population that seems to be recovering. So it's now, it's kind of out on a peninsula, so it's an isolated population, and it now seems to have devil-free individuals, or tumor-free individuals, and an increase in numbers.
Bye.
That's what their teeth look like. They're actually really calm, at least this one was, when they're being handled. It sits there and lets them out. Okay, so like I said, this is a big international collaborators. People in my lab and multiple other institutions and funding sources. That's it.
Go.
So did you say what the effective size of some of these populations are? Like how small are they? Are they small?
Dozens of individuals.
So really small.
Yeah, 20 or 30.
Any idea of the total number across Tasmania?
That's a good question, and I'm not sure. I can't remember, but the disease has knocked the total population down by like 80%.
Like hundreds, thousands,
Thousands.
just thousands.
Yeah. But I don't have the guess.
In the capture interactive protocol, do you have to account for any kind of bias for outlier detection? Or intentionally capturing candidate genes and that kind of thing?
In terms of, yeah, basically. So you're starting out with a set of candidates instead of just an anonymous scan across the genome.
Right.
So you're right, it probably wouldn't be appropriate to take rapture data and just plug it into a standard outlier detection model.
Can you help me understand? You say you want to have one loci for every scaffold.
Mm-hmm.
If you have 15,000, you said there were 30,000 scaffolds. Yeah.
So that was mostly, so a subset of those loci we wanted to just have well spread across the genome. And so one way to do that was not to have more than one per scaffold. Does that make sense?
So many scaffolds did not have any. You're saying you just did the rule of no scaffold did not run.
Yeah. Yeah, so lots of scaffolds didn't have any in that array. And the way this genome assembly works, the scaffolds are assigned to chromosomes. These guys only have 6 chromosomes, so they're really big. So all the scaffolds are assigned to chromosomes, but the order on those chromosomes is not very well known.
I have another question about minor allele frequency cutoffs. So is this a scenario where you might want to really look for rare variants or particularly changes in really rare variants over time?
Yeah, that's a good question. So Yes, we did our minor allele frequency cutoffs on the whole data set, so if there's something that started out really rare and became more common, it should still pass the filter. We're likely only to be able to detect big effect things in general. And so if something goes from 2% to 4%, we're not going to be able to detect it. It's probably not a big effect. If there would be any way to detect it.
And did you only use that cutoff in your neutral loci, or did you use it in everything?
I think we had a less stringent minor allele frequency cutoff for the other loci. I can't remember, 0.1 or 0.05. And then for the neutral ones where we just wanted to scatter across the genome. All right, so I don't know when dinner is, but I can keep helping out with stacks if you guys want to keep hammering away, or